Welcome to our exploration of the evolution of genomic editing. In this video, we'll journey through the fascinating history and breakthroughs that have shaped how we understand and manipulate genetic material. From early discoveries in DNA structure to the revolutionary tools of today, genomic editing has transformed science, medicine, and agriculture. Let's begin by setting the stage for what genomic editing means and why it's so significant in our modern world. Story of genomic editing starts with the discovery of DNA's double helix structure in 1953 by James Watson and Francis Crick. This breakthrough revealed the blueprint of life and opened the door to understanding how genetic information is stored and transmitted. The double helix model provided a foundation for future research, setting the stage for scientists to explore how genes could be identified, isolated, and eventually edited. The decades following the discovery of DNA's structure, scientists began experimenting with ways to manipulate genetic material. Early methods involved using chemicals or radiation to induce mutations, but these approaches were unpredictable and often resulted in unintended consequences. Despite these challenges, these early experiments laid the groundwork for more precise techniques that would emerge later. 1970s marked a turning point with the development of recombinant DNA technology. Scientists discovered how to cut and paste DNA from different organisms using restriction enzymes and ligases. This allowed for the creation of genetically modified organisms and the production of important proteins like insulin. Recombinant DNA technology was a major leap forward, making genetic engineering more precise and opening up new possibilities in medicine and agriculture. The 1980s, the invention of the polymerase chain reaction, or PCR, revolutionized genetic research. PCR made it possible to amplify small segments of DNA, enabling scientists to study genes in greater detail and perform genetic tests with unprecedented accuracy. This technique became a cornerstone of molecular biology and paved the way for advances in genomic editing. Late 1980s saw the development of gene targeting techniques in mice, allowing scientists to create knockout models by disabling specific genes. These models became invaluable for studying gene function and disease, providing insights that were previously impossible to obtain. Gene targeting in mice marked a significant step toward more precise and controlled genetic modifications. The 1990s, zinc finger nucleases, or ZFNs, emerged as one of the first targeted genomic editing tools. ZFNs are engineered proteins that can recognize and cut specific DNA sequences, allowing for targeted gene modifications. While powerful, ZFNs were complex to design and limited in their flexibility, but they represented a major advance in the quest for precise genome editing. Shortly after ZFNs, scientists developed transcription activator-like effector nucleases, or TALENs. These tools offered greater flexibility and specificity in targeting DNA sequences. TALENs made it easier to edit genes in a variety of organisms and further expanded the possibilities for genetic engineering. Their development marked another step forward in the evolution of genomic editing technology. Early 2010s brought a true revolution with the discovery and adaptation of CRISPR-Cas9 for genome editing. CRISPR, originally a bacterial immune system, was harnessed as a powerful tool for making precise cuts in DNA. Its simplicity, efficiency, and versatility made it accessible to labs around the world, sparking an explosion of research and applications in medicine, agriculture, and beyond. CRISPR works by using a guide RNA to direct the Cas9 enzyme to a specific DNA sequence. Once there, Cas9 makes a precise cut, allowing scientists to add, remove, or alter genetic material. This method is not only faster and cheaper than previous techniques, but also highly accurate, making it the tool of choice for many genetic engineering projects today. Genomic editing has opened up new possibilities in medicine. Scientists are now able to correct genetic mutations that cause diseases, develop gene therapies, and even engineer immune cells to fight cancer. These advances are bringing us closer to personalized medicine, where treatments can be tailored to an individual's genetic makeup. In agriculture, genomic editing is being used to create crops that are more resistant to pests, diseases, and environmental stresses. Scientists can now develop plants with improved nutritional content, longer shelf life, and better yields. These innovations have the potential to address food security challenges and promote sustainable farming practices worldwide. With the power to edit genomes comes significant ethical responsibility. Questions have arisen about the potential for unintended consequences, the possibility of designer bobbies, and the impact on biodiversity. Scientists, policymakers, and the public must work together to ensure that genomic editing is used safely and ethically, balancing innovation with caution. As genomic editing technologies advance, governments and international organizations are developing regulations to guide their use. These frameworks aim to protect public health, ensure safety, and address ethical concerns. The regulatory landscape is evolving alongside the technology, requiring ongoing dialogue and adaptation. Public perception of genomic editing varies widely. While many people are excited about the potential benefits, others are concerned about safety, ethics, and the unknowns. Education and transparent communication are essential to building trust and ensuring that society can make informed decisions about the use of these powerful technologies. One of the most controversial applications of genomic editing is the development of gene drives, which can spread genetic changes rapidly through populations. While gene drives could help control pests or eradicate diseases like malaria, they also raise concerns about unintended ecological impacts. Careful research and regulation are needed before deploying such powerful tools in the wild. Genomic editing can target either somatic cells, which affect only the treated individual, or germline cells, which can pass changes to future generations. While somatic editing is generally accepted for treating diseases, germline editing is far more controversial due to its heritable nature and potential long-term effects on the human gene pool. Recent advances have led to the development of base editing and prime editing, which allow for even more precise changes to DNA. These techniques can correct single-letter mutations without making double-stranded breaks, reducing the risk of unintended effects. Base and prime editing are expanding the toolkit available to scientists, offering new hope for treating genetic diseases. Gene therapy, powered by genomic editing, has already achieved remarkable successes. Patients with inherited disorders like sickle cell anemia and certain forms of blindness have seen life-changing improvements thanks to targeted gene corrections. These stories highlight the real-world impact of genomic editing and its potential to transform lives. Genomic editing isn't limited to humans and plants. Scientists are also editing animal genomes to improve livestock health, create disease-resistant species, and even revive extinct animals. These applications raise their own set of ethical and ecological questions, but they also offer exciting possibilities for conservation and food production. Genomic editing is a key driver of synthetic biology, a field that aims to design and build new biological systems from the ground up. By combining engineering principles with biology, scientists are creating organisms with novel functions, from bacteria that produce biofuels to yeast that synthesize medicines. Synthetic biology is pushing the boundaries of what's possible in life sciences. The ability to edit genomes is paving the way for personalized medicine, where treatments are tailored to an individual's unique genetic profile. By understanding the genetic basis of diseases, doctors can develop targeted therapies that are more effective and have fewer side effects. Personalized medicine represents a major shift in how we approach healthcare. Rapid progress in genomic editing has been fueled by global collaboration among scientists, institutions, and countries. Sharing data, resources, and expertise has accelerated discoveries and helped establish best practices. International cooperation is essential for addressing the complex challenges and opportunities presented by genomic editing. Despite its promise, genomic editing faces significant challenges. Off-target effects, where unintended parts of the genome are altered, remain a concern. Technical limitations, high costs, and regulatory hurdles can also slow progress. Ongoing research is focused on making genomic editing safer, more efficient, and accessible to all. Educating the public about genomic editing is crucial for informed decision-making. Outreach programs, science communication, and transparent reporting help demystify the technology and address misconceptions. By fostering understanding, we can ensure that genomic editing is used responsibly and for the benefit of society. Looking ahead, the future of genomic editing is full of promise. Advances in technology are making editing more precise, efficient, and affordable. As our understanding of genetics deepens, new applications will emerge, from curing rare diseases to enhancing crop resilience. The possibilities are limited only by our imagination and our commitment to ethical science. Artificial intelligence is playing an increasingly important role in genomic editing. AI algorithms can analyze vast amounts of genetic data, predict the effects of edits, and design more effective editing strategies. By integrating AI with genomic technologies, scientists are accelerating discoveries and improving outcomes. Open science initiatives are making genomic data and editing tools more accessible to researchers worldwide. By sharing information openly, the scientific community can collaborate more effectively, avoid duplication of effort, and accelerate progress. Open data is helping to democratize genomic editing and ensure that its benefits are widely shared. Bioinformatics is essential for analyzing and interpreting the massive amounts of data generated by genomic editing. Advanced software tools help scientists identify target genes, predict outcomes, and monitor for unintended effects. The integration of bioinformatics with laboratory techniques is streamlining the entire editing process. International organizations are working to establish guidelines for the safe and ethical use of genomic editing. These guidelines help harmonize standards across countries, promote responsible research, and protect human rights. Global cooperation is key to ensuring that genomic editing benefits all of humanity. Citizen science projects are involving the public in genomic research. By contributing data, participating in experiments, or helping analyze results, non-scientists can play a role in advancing genomic editing. These initiatives foster engagement, transparency, and a broader understanding of the science. Genomic editing is being used to address environmental challenges, such as developing plants that can clean up pollutants or engineering microbes to break down plastic waste. These applications demonstrate the potential of genomic editing to contribute to environmental sustainability and restoration. As genomic editing becomes more powerful, scientists are developing biocontainment strategies to prevent edited organisms from causing unintended harm. These include genetic safeguards, physical barriers, and monitoring systems. Biocontainment is essential for ensuring the safe use of genomic technologies in the environment. Possibility of using genomic editing for human enhancement, such as increasing intelligence or physical abilities, has sparked intense debate. While some see potential benefits, others worry about inequality, safety, and the definition of what it means to be human. Society must carefully consider the implications before pursuing such applications. As new genomic editing tools are developed, questions about intellectual property and patent rights have emerged. Balancing innovation with access is a challenge, as patents can both incentivize research and restrict the use of important technologies. Ongoing legal and policy discussions will shape the future of genomic editing. With the rise of genomic editing and personalized medicine, protecting genetic data privacy is more important than ever. Safeguarding sensitive information from misuse or discrimination is a top priority for researchers and policymakers. Robust data protection measures are essential for maintaining public trust. Funding from governments, private companies, and philanthropic organizations has been crucial for advancing genomic editing research. Investments support basic science, technology development, and the translation of discoveries into real-world applications. Continued funding will be essential for unlocking the full potential of genomic editing. Preparing the next generation of scientists is vital for the future of genomic editing. Education programs, hands-on training, and mentorship help build the skills needed to advance the field. By investing in education, we ensure that future breakthroughs will continue to drive progress. Women have played a pivotal role in the evolution of genomic editing, from foundational research to leading cutting-edge projects today. Promoting diversity and inclusion in science not only ensures equal opportunities but also enriches the field with a broader range of perspectives and ideas. Genomic editing has the potential to transform public health by enabling rapid responses to emerging diseases, improving diagnostics, and developing new vaccines. These capabilities are especially important in the face of global health challenges such as pandemics and antibiotic resistance. Perhaps the most exciting promise of genomic editing is the potential to cure previously untreatable diseases. As research advances, more conditions may become manageable or even curable, offering hope to millions of people worldwide. The journey from discovery to cure is ongoing, but the progress so far is truly inspiring. The evolution of genomic editing is reshaping society in profound ways. From changing how we think about health and disease to raising new ethical and legal questions, the impact of this technology extends far beyond the laboratory. Society must continue to engage in thoughtful dialogue as we navigate this new era. As we look to the future, the possibilities for genomic editing are vast and exciting. Continued innovation, responsible stewardship, and global cooperation will be key to realizing the full benefits of this transformative technology. The story of genomic editing is still being written, and its next chapters promise to be just as groundbreaking as those that came before. Let's recap the key milestones in the evolution of genomic editing. The discovery of DNA's structure, the advent of recombinant DNA, the development of PCR, the rise of targeted editing tools like ZFNs, TALENs, and CRISPR, and the emergence of base and prime editing. Each breakthrough has brought us closer to unlocking the full potential of the genome. Evolution of genomic editing is one of the most remarkable scientific journeys of our time. From humble beginnings to groundbreaking innovations, this field continues to push the boundaries of what's possible. As we move forward, it's up to all of us, scientists, policymakers, and the public, to ensure that genomic editing is used wisely, ethically, and for the benefit of all.